Well, good morning. I'm Jennifer Whitaker. I'm the Director of Talent Management, and I want to thank you for joining us today for Sunrise Science Lecture Series. We're excited to have you all with us this morning, and even though things look a little bit different, normally we're gathered in Fishman Auditorium and we're having breakfast and joining one another in person. We're doing things a little bit differently. And we're coming to you virtually from our offices and from our homes as you are joining us as well. So for those of you who may be new to the Sunrise Science Lecture Series, this is an informal lecture series. And the intent is for us to highlight different areas of research that are happening around the Institute. And although the presentations are designed to focus on scientific work, the event is open to all members of the SVP community and really with the delivery in mind to be understood by a general audience. So just as in the past, we hope to have time at the end of the presentation where we can have questions to be addressed by our lecturer. And there is a Q&A feature at the bottom of your Zoom window. Hopefully you can locate that now. And you can type any question through that Q&A feature and submit that to us. We're going to hold those questions till the end of the presentation, but certainly feel free to submit them at any time during the presentation as questions come up that you want to know more about. So I have the pleasure of introducing our lecturer today, Dr. Ze'ev Ronai. Dr. Ronai is a professor in our Tumor Initiation and Maintenance Program, as well as in our NCI Designated Cancer Center, and serves as the Institute's Chief Scientific Advisor. Dr. Ronai completed his undergraduate degree in biology, his master's in microbiology, and his PhD in immunobiology at the Hebrew University in Jerusalem, Israel. He completed postdoctoral training in molecular biology at Columbia University in New York. Following his postdoctoral studies, he stayed on the East Coast and he joined the faculty at New York Medical College, where he established the Molecular Carcinogenesis program. He later moved to the Ruttenberg Cancer Center at Mount Sinai School of Medicine, where he was tenured professor. And then in 2004, Dr. Ronai came to Sanford Burnham Prebys. During his time at Sanford Burnham Prebys, Dr. Ronai has held various leadership positions, which have included the Director of Signal Transduction Program, the Deputy Director of the NCI Cancer Center, Scientific Director, and currently our Chief Scientific Advisor. I'm so glad that Dr. Ronai agreed to speak with us today. And so without further ado, please welcome Dr. Ze'ev Ronai.
Thanks, Jen. So it's a pleasure and honor to speak to you and describe to you what is actually that we do, how we think, what motivates us, and some examples, 3 or 4 examples of studies we have done over the last few years. And I have to apologize to the scientists that listen to this presentation because Sunrise for me is a presentation to our administrators so that are able to follow us and understand what it is that we're about. I'll start with those that actually do all the work, and I won't be able to speak to you without them. They are the ones that kick it hard day and night. Hangsu, Erez, Marzia, Ali, Sachin, Ben, Stefania, Young May, and Regina that helps all around. So the first point that I would like to highlight in this presentation, there'll be a few of those introduction slides that will gear up to actual studies that we have done in recent years. And the first is what is really important to us. So I would summarize what's important as by indicating that we want to make valuable, impactful discoveries. And impactful can be measured by two ways. One, moving the field forward scientifically. And the second is paving the road for clinical trial of clinical evaluation. And over the years, more and more of our studies managed to go to the direction, and I'll explain to you and share with you a few of those examples. Why we do what we do? What is the motivation? What drives us to work so hard? So if we would be face-to-face sitting in Fishman together, as usually is the case, I would ask you to answer, who are those ladies that you see here in this slide? And given that we are zooming in, I'll have to provide you the answer. I assume that there'll be very few of you that will be able to tell me anything else beside they look nice. And indeed, the lady on the left, Becky, is the lady on the right here. Becky Derman has been Miss America in 1974. And her daughter, Diana, has been Miss Colorado in 2011. Why I bring it up here? Because Becky, I know personally from the board of Melanoma Research Foundation. And the reason I know her is because Becky was diagnosed with melanoma in 2010, fought it through all the hard lines one can imagine, managed to be a survival that speaks very well for what she went through, and a very good motivator to understand what is about and why it is that we're doing what we're doing. So it is the inspiration from cancer survivors and the drive by clinical data that we are currently able to achieve or to obtain that drives our science that is meant to solve clinical problems. So next point that I will shortly highlight briefly is how we decide to work on, what to work on. And I would emphasize the word usually because there are exceptions to any rule, and I'll give you examples of the rule and the exception. So every day we come up with the great ideas. Every day we have an idea how we can solve a problem, hypothesis that is wonderful, but we are not able to do everything we think about. So we need to filter them and select those that are more amenable or realistic. So if we have a great idea, we next ask whether it will be relevant to a patient population. That by itself will limit the number of problems that we want to solve. Then we ask, do we have the tools? And tools are, do we have the technology? Do we have the know-how? Do we have the expertise? And answering this question further filter down the number of problems that we can address. Then we ask, well, if we take it, address it, solve it, work on it for the next few years, would it impact the greater community? And answering this question further filter down the number of questions that we would like to ask. And finally, the maker or breaker is, do we have a trooper in the lab? Do we have a person in the lab that would be excited by this problem and will be willing to drive it, to try to solve it over the next few years? And this, by default, reduces the number of options that we can consider working on significantly. Couple of words about cancer, the genetic and the epigenetic network, and the rewiring, which is the title of this presentation, and then we go for examples. There are multiple types of any given cancer. There are multiple types of breast cancer, multiple types of lung cancer, melanoma, and so on and so forth. And we are able to distinguish between those types based on genetic information and based on epigenetic information. Genetic being the mutation, the hardcore fixed elements, and epigenetics, largely RNA protein. Those we're related to in a minute because my lab has largely been focusing on the epigenetic trait. And these two together dictates whether tumor will metastasize, whether tumor will resist therapy, or will respond to select drug, and what will be the drug it will respond to. A little more about that, the genetic events, which are the mutations, can be inherited. We can get it as a germline mutation, or they can accumulate during our life. We call them somatic mutations, and they will accumulate because we exposed ourselves to too much sunlight, or we smoke, and these are the known factors. But the same factors in sunlight, smoke, diet also have a big impact on the epigenetic milieu. These are changes driven largely by the environment. They are also temporarily, they're not fixed, but they are very important in driving tumor ability to resist therapy, to develop metastases, and to change or be unique out of the big mess of the tumor that lies within. And the epigenetic maps that we learned how to build and we learned how to read over the last decade provide much more accurate information than just the genetic. And we in the lab and many others focus on understanding the genetic maps and understanding how to break them apart. So this is a very, very simple diagram that explains why cancer is smarter and what is the base for the rewiring doctrine. So normal cells proliferate. If it face a problem, it has a way to decide whether to live, overcome the problem, or to die. But the tumor cells accumulate mutations. The first mutation is not sufficient to develop tumor. And a good example of that is the nevi that we have on our skin in our body. These are the nevi that 95% of those nevi have actually mutation that is seen in most melanomas. But they will not go anywhere and they will not do anything until additional mutations are going to occur because of exposure to additional carcinogens, sun rays, and so on and so forth. But these are not only mutations, there are also epigenetic changes that accumulate in time. And each one of those events will change the features and the nature of the tumor so that it allows now to escape immune system, to escape death cues, to grow faster, to go to sleep, to become dormant, or to metastasize. And one of the ways we can monitor these, especially in tumors that we can actually see, those are the skin tumors, is simply to monitor what is that happen to us in our body. So as you know, much of the research in my laboratory is focusing on melanoma, and in melanoma, we do have a very interesting campaign that's called Get Naked, as you can see here. And the idea behind that is that we just check ourselves and see whether those moles that we have in our body have grown, have changed colors. And that basically is the first indication that we need to be aware and something need to be attended to. So the notion here, there are multiple events, genetic and epigenetic, that takes place in a cell, and they are needed in order to acquire the cell ability to become smarter, to escape, to metastasize, and so on and so forth. So what are all this rewiring? Here's a way by which I like to explain what rewiring means. We are in San Diego and we want to drive to Los Angeles. And we know that we will be able to do it on Route 5 going north. That's a simple way, 545, and we make it. The tumor is smarter. The tumor decided that in order to get to Los Angeles, it can go through Catalina, through the sea, or through the east Route 15 and other routes in order to get to Los Angeles. And this is the rewiring that we need to figure out in the work that we do in the lab. What happened and how the tumor decided to make a left turn, right turn, up, down in order to disobey the normal route and to acquire the features that it does have. So years ago, when we published our first study that had to do with rewiring in melanoma, this is like 13 or 14 years ago, we came up with this diagram, and you can see here the dials of a clock, and each dial have a number gene, important gene that drive melanoma. But if you can pay attention, you see that these dials will never make this clock work. And I remember that we came up with this image, and Guy Salveson came to me and was pretty upset. I mean, this will never work. You know, this is the whole purpose of highlighting how the tumor is able to do what we think is not going to work, actually work. And what we are doing, as I mentioned, is figuring out the dials and what to make out of them. All right, so let's start with examples of work that we have been doing and what do they mean. The first problem I would like to discuss with you is the problem of cancer escape from the immune system. So usually tumors develop under the radar of the immune system. So immune system does not sense them, does not fight them, and tumor evolve and grow. And we recognize it for years. We have different names for that. And we have been trying to challenge the tumors so that we force them to be seen by the immune system, but not very successfully. A little bit here, a little bit there, but not very successfully. So many tumors are not seen by the immune system, and they are considered to be cold because they even don't respond to immune therapy. And immune therapy that became a highlight in recent years is a principle that I'm going to refer to again and again today, is the notion that we're using tricks against our immune system so that the immune system break loose. And in doing so, the immune system attack the tumor, but also at the same time attack ourselves. And I'm going to relate to that in a few minutes. Here the question is, can we turn a cold tumor to become a hot one? And the example that I'm going to give you from study, actually this is the more recent published study from our laboratory, and the scientists can read. Those that follow me got to understand my accent by now, are able to follow the picture, and I'll explain the picture and relate to the picture. And what you see are chameleons that are hidden, blended very well with the colors, and are not seen by the birds. Chameleons are referred to by analogy to the tumors, and the birds for the components in the immune system that's supposed to detect those and take care of them. Can we make these green chameleon turn colors stronger and stronger so that they will be detected by the immune system, by the birds that will eliminate them? This is the challenge. So we have taken upon and managed to identify a component that is able to make this green purple. And in doing so, make the tumor seen by the immune system. This is an example for a hypothesis-based study, because the protein, the components that we followed here could be identified to be expressed at high levels or present at high level in tumors. And when present at high level in tumors, it was a bad prognosis. Tumors did not do well. So we knew that we wish to possibly inhibit this guy. And since this was also found to be highly related to failure to respond to immune system, we reasoned that this guy, this protein we study here, may have an implication on the immune system. So to make a long story short, we were able to take tumors in which we inhibited this component, and we found that when we inhibited the component, one gene in the tumor was suddenly seen by the immune system and was removed by the immune system, or was blocked in terms of growth by the immune system. But if we have done the same experiment in an animal that did not have intact immune system, the tumor kept on growing and nothing happened, which to us was a very important sign that the immune system is actually the required component that will eliminate and recognize and eliminate these tumors. So we mapped exactly what are the components in the pathway that are regulated by this protein, and I'm not going to relate to that. I apologize to the scientists here. And we were able to further demonstrate that in doing so, we can take this cold and make them hot tumors. The good thing about this is the guy we are inhibiting here, the protein we inhibit here, actually has been on target by the pharmaceutical companies. And the three pharmaceutical companies have developed actually drugs that inhibit this guy. So in theory, we are able to go relatively fast to clinical trials where we can test whether inhibition of this guy would be helpful to eliminate tumors that at present do not respond and are escape the immune system. The work that I just described to you was led by Hang-Soo Kim, a talented rep in our lab, about 2.5 years of hard work. And the outcome of this study is currently being translated to a clinical trial that is being about to start, I hope, in 2021 at MGH, Harvard, together with GSK, where 7 different tumors that are considered cold will be subject to examination with this inhibitor and immunotherapy. This is one example for a study that had a rationale and had an hypothesis we could follow because we had the data from patients that allowed us to reason to go and to test the hypothesis. But not always this is the case. And the example that I provide you here, the magician, can be also considered science by serendipity. And those of us that tell you that we know everything that we do and we follow everything to the letter because our hypothesis turned out correct, that will not be the case. That serendipity does play a very important role in our discoveries and dictate many discoveries in turning the page into a direction that we usually do not expect. And here is one such story. The reason this was not hypothesis driven is because not all genes or proteins we study in the lab can be looked at based on clinical information. And there is many technical reasons for that, but I'll stay with that. It's not always possible to do that initial examination. And the protein at stake here is called RNA5. It's a very small molecular weight, small protein. It is involved in the control and quality control of proteins in the cells. In other words, cells have a built-in machinery that maintain the quality control. And if something goes bad, this guy turned on. And then there are 2 options that things could happen. 1, quality control fix and cells continue. 2nd, quality control cannot fix, cells undergo cell death or other processes. And the magician here, as you can see with the wand, dictates what are the cells that will fly out of the head, and those are immune cells. And as you see, the cells change their shape, or there are more and more specific type of cells that proliferate, or more of them are seen as they come close to the tumor. And this is the discovery we made here, a very specific effect on immune cells that can attack the tumor. How we came about it. So we studied this gene for more than a decade. We did a lot of cell biology, biochemistry, interesting discoveries, and we even generated the mice that lacks this gene, knockout mice. And for over a decade, we held the mice. We considered it the boring mice in the lab, simply because it didn't show us anything that is unusual. Up to the point that a talented postdoc in the lab decided to put tumor cells into these animals. And she found that mice lacking this RNA5 gene did not allow the growth of tumors. So absence of one gene in this animal was sufficient to inhibit melanoma growth and also seen for other tumors, including colon cancer. The other thing that was part of the first surprise is that these animals inhibited the tumor growth because they have developed a very strong immune response against the tumor. So suddenly, an animal that did not appear to have any problem, appeared to be healthy, we had it over a decade, just implementation or inoculation of small amount of tumor cells, they did not develop because they were effectively attacked by the immune system. So that was the first surprise. The second surprise was that it's all that I just told you about is driven by the gut microbiome. Totally unexpected. A long story how we made this observation, but it is the gut microbiota that affect the strong anti-tumor effect, anti-tumor immunity. Not only that, but we were able to break down the components in the gut microbiome and identify what are the bacterial strains that are sufficient to mediate this anti-tumor immunity, identifying 11 of them that just inoculation of those was sufficient to turn the immune system and to inhibit tumor growth in animals. So what do you do with that? So this work was done by Yan Li, day and night over a 4-year period. And this story, this study required expertise that we did not have in the lab at the time: tumor immunology, gut microbiome, computation of gut microbiota, and extensive clinical collaborators. So this study had over 40 collaborators that helped us understand what is that we see in the immunology part, Linda here, together with Roberto at the time, the gut microbiome, Scott Peterson, and external collaborator Amanda in Nebraska, computation of microbiota with Tao Long, and clinical collaborators from 3 different places around the world. And with those together, we were able to make the notion that, number one, gut microbiome control anti-tumor immunity, identifying precisely what are the bacterial components that do so. And RNA5, this is the gene we study all these, along all this, control both the gut microbiota and the anti-tumor immunity. And it does so by its role in the quality control in the cell. Not only that, but the components of the quality control we found can be used to predict if patients will or will not respond to immunotherapy. So where this goes, if we have identified 11 bacterial strains that are sufficient to turn anti-tumor immunity and inhibit tumor growth, can we identify fiber or alike that will induce or enrich those bacterial strains and induce anti-tumor immunity? That has been one of the questions that we asked. And I want to just note that prebiotics are food components, and I mentioned fiber as one example, that will affect the composition of the gut microbiota, will cause enrichment of some bacterial strains over the other, as opposed to probiotic, which is already a mix of a given bacteria strains that we're having in our yogurts or whatever else we do. So we are shying away from probiotics and we are focusing on the prebiotics as a potential a component that will change the milieu of the gut microbiota and help fight the cancer. So in the work that was following up, the one I just mentioned to you, we found 2 prebiotics, inulin and mucin, and either one of those was found to induce anti-tumor immunity and to limit melanoma growth. One of them we administered just in the food of the mice. The other was provided in the water that the mice drank, and either one of them drank or ate these for a couple of weeks, inoculated with tumor cells, and the tumor did not do well, pointing to the anti-tumor immunity that we have mapped in these animals. So this study was published a few months ago, and one of the take-home messages that I learned to highlight together with mentioning the study is, please don't think about using these prebiotics in any way, shape, and form. This is too early in the game, and I can talk to you much more about that, but I have to emphasize this as I describe this study, which is exciting for us because it does take us an important step forward for possible practical implications of what is that we are doing with these gut microbiome studies. Well, as you know, each coin has 2 sides. And I just told you about the mice that is able to very effectively fight tumors by having strong anti-tumor immunity. I also told you that breaking the immune system, as we do with immune checkpoint therapy in the clinic nowadays, does result in extensive side effects, because when we break the immune system attacks not only the cancer, but also ourselves on tissues. And so we ask whether the mice that I've just described to you, those brave mice that fight tumors, those boring mice that did not show us any phenotype, could also have an indication of autoimmunity under some conditions. And we found that this is indeed the case, and we did not need to do much in order to see this autoimmunity. What we found that if we take these animals and we add a little bit of an inflammatory agent to their drinking water, nothing else, they are going to develop very strong intestinal inflammation, very severe symptoms that are considered similar to IBD, which is common in men. And we found what is the mechanism, how this works. You break loose the key, you allow inflammation to take place, and this inflammation results in inflammatory disorders. So mice that lack this RNA5 gene not only fight tumors, but they have also severe IBD symptoms, intestinal inflammation. We were able to identify why, what is the components that drive this. This is one of the key components we identified, and we found that there are antibodies that people have generated against this guy. Use these antibodies and were able to block the formation of IBD in these animals. So we are able to halt the inflammatory disorders if we prevent or neutralize one component only. This is mice. We want to go to men, and we are collaborating at the moment with Adriano Agussi in the University of Zurich, who has a system where he screens human antibodies against given antigens. And this is one of those that he has been helping us identify human antibody, and it looks like we are getting there. So this is promising. I'm going to move to a totally different story. And again, I'm going to describe the story through the image that you see in front of you. This is classic fires in California. We're all familiar with that. And you see 3 airplanes. You see one airplane that is trying to cover the fire and is doing a somewhat good job, but not enough. You have another airplane that drops another chemical on the fire and makes the fire worse. You have a third airplane that has the combination of the two and is able to eliminate the fire. So takeoff message, there is some combination drug here that I'm going to talk to you about, which we have identified, which are able to turn off the fire, inhibit the tumor. So what is the story? The story focuses on one protein or enzyme, a metabolic enzyme. It's called asparagine synthase. But to those of us that don't need to go too far in understanding what the story is about, I will say that this metabolic enzyme is very important for cancer growth. And if you don't have it, cancer will suffer. The problem is, and a very important point that I need to emphasize, is that the level of this enzyme is low in both hematological and solid tumors. So to start with, this is a good sign, because if an enzyme level is low, there is an opportunity to target it, opportunity for therapy. And along those lines, this has been actually explored, and it has been explored more than a decade ago, where FDA-approved drug against the SNS, called LAs, was found to be effective therapy in certain hematologic malignancies. ALL is one example. So even today, a patient that would have ALL could get LAs as part of their therapy, and it works. But it does not work in solid cancer. It doesn't work in pancreatic cancer, melanoma, colon cancer, and so on and so forth. So we set ourselves to try and figure out why not. And if we figure out why not, can we tackle it? Can we solve it? So the work was led by a talented postdoc in the lab, Gaurav Patria, collaboration with a genius in computer science, Eitan Rupin at NCI. And the way we performed the study was trying to figure out what has changed in solid tumors when we inhibit this specific enzyme. And we noticed that, again, along the quality control pathway, there is a remarkable change. But the computer science here came to our aid in order to find what could be the partner for this enzyme that will eliminate the tumor. And today, with a proper computational power, one can go and identify what will be the synthetic lethality, or what is the partner of two genes when combined will be able to effectively eliminate cancer. And this is the work that Eitan contributed. We gave him the component SNS, and we asked what will partner with this enzyme to eliminate pancreatic cancer. Eitan came back to us and told us, gave us a list of about 6 different components along the same pathway. And that made a lot of sense to us, interrogated by Gaurav, found to be correct. And the bottom line is, indeed, combination of 2 components would be sufficient to make solid tumors succumb to this therapy. And this is exactly what we managed to do, prove synthetic lethal partnership by the combination of the ASNS inhibitor and the MAP kinase inhibitor, 2 different components that can be used for effective therapy in pancreatic cancer as well as in melanoma. And we have published this work just a few months ago, and this work is currently being developed to a clinical trial at the Oregon State or Health Science Center with a talented oncologist in collaboration with a company that will provide the drugs. And so I'm hoping that we would have a new way to address possible solution for the pancreatic cancer, which is a big problem for us all. I will close with one last brief story. And the story has to do with the rabbit and the hare. And you can see here, you can see a race between the turtle and the rabbit. And you can see that in this race, something happened to the rabbit. You see the word Glenn written on the rabbit, which means there is very high level of an amino acid called glutamine. But you also see a tumor in the rabbit that basically did not work well because the one who made this race was the turtle and not the rabbit. So this is a story, and in brief, and what we have done here, and this is the complete picture, we found that high level of glutamine in breast cancer are the reason that breast cancer will not respond to common therapy like paclitaxel. We explained the mechanism and why it is working like that, and we focused and demonstrated that there is a cluster of breast cancer, triple negative breast cancer patient, that if they have high level of glutamine, one would like to reduce it before administering paclitaxel in order to have effective therapy. One word about the image. So we prepared the image when our studies accepted publication in order to try to make a cover of an issue. Not always we succeed. But here I was upset to some degree because we didn't make the cover. It's a remarkable image. And it was my wife that needed to remind me that A, you don't put tumors in rabbits, and B, you do not break myth or stories that kids love, which is the rabbit and the hare race. And we have disobeyed these two, and the result is we have a great image that can explain the story, but this is it. So high level of glutamine in breast cancer patient block the effect of paclitaxel. Therefore, you want to inhibit glutamine in order to have effective paclitaxel treatment. And this is seen in about 30% of triple negative breast cancer patients. The result of that was that a company in South San Francisco that generated glutaminase inhibitor, CB839, picked up on our study and decided to go into clinical trial without us knowing anything about that. And it was a phone call that I got a year later in which the lead of the company thanked me for our ability to guide them to do the right type of study clinically, where they have administered paclitaxel together with their drug and managed to take care of triple negative breast cancer that have high level of glutamine. So this is one of those nice stories that you even don't escort in terms of development, but it does have meaningful outcome. This is a multi-center clinical trial that is running not only in the United States, but also in Europe nowadays. Youngju, who did join the lab and decided that she works on breast cancer, did all the work. She got a very strong support from clinical collaborators that helped establish that triple negative breast cancer would be the one to pay attention in this line of studies. All right, so I'm going to wrap up. What I told you today was that we tackle difficult problems that are relevant to clinical unmet needs. In most cases, these are hypothesis-driven. They are always patient-justified, but at times serendipity does take over. Some of our studies reach clinical evaluation. This is the ultimate goal that we set ourselves. All this takes place thanks to talented group of lab members who devote tireless efforts to make science stick. And when I say tireless effort, guys, I do mean it. Being at SVP provide the right infrastructure, environment, and support, and this is a key to drive fast and be able to reach high. None of this would be successful without the support from primarily NCI. I'm blessed by support from NCI, DOD, and Melanoma Research Foundation, Melanoma Research Alliance, and others. And I'll finish by showing this picture. Then this is the picture of the scientific family that I've grown up over the years. Once every 4 or 5 years, we are performing a scientific retreat to which we invite alumni that come from all over the world to be part of this. The last one was last summer. So this is pre-COVID, and examples is Pablo from Argentina, Youngju from Korea, Junfei from Maryland, Serge from Pennsylvania, and we have quite a few other, Eric from the Moffitt in Florida, and so on and so forth. We do invite scientists that attend these events and help us be honest or shape up further. David Fisher, head of dermatology at MGH, and Eitan Rupin were those that were part of these events. And you all know Annie, who also was part of this retreat. I'll stop here and I'll be delighted to answer any questions. Thank you all.
Thank you, Ze'ev. That was a great presentation. I think we'll be thinking about that story of the tortoise and the hare a little bit differently now. I appreciate that. So we have opened it up for questions. So if you do have any questions, you can use the Q&A button down at the bottom of the screen. And Ze'ev will be staying with us for a few minutes. So if you can submit those there, we will take questions and share them for everyone to hear, and then for Dr. Ronai to respond. So I did have a question, Dr. Ronai, if you could answer this. I think this is quite interesting. As I was thinking about how your lab works on many different kinds of cancer, when you start, do you start with an interest in applying research to a specific tumor that's unique to a specific cancer type? Or is it something that's discovered after reviewing results of some of the research that you have?
Yeah, so actually it's a very good point or question, because years ago we would take a a problem and tackle it just because it is interesting. We no longer do that. When we have an interesting question, we first go and assess whether it will be relevant in a given patient group. And more than once over the last few years, we started to work on a problem in one cancer, and we shifted to another cancer because we realized that the relevance would be in a different type of tumors. So Ali in the lab that started to work on melanoma a few years ago shifted to work on AML because we found that the gene that he studied would be much more relevant to AML. And Ben, a student in the lab that started to work on melanoma, shifted to prostate cancer just because we found that it would be much more relevant to address the question in prostate cancer. This is just two recent examples, but many more of them exist. And yeah, we are driven by what the clinic, what the patient data actually tells us.
So we have another question that has come in, and the question states, it is clear that many of these discoveries are leading to new anti-tumor therapies. Do any of your discoveries also show promise for early detection of cancer?
This is a very important point, and ultimately that's where we all want to go. We want to be in a position that we'll be able to detect cancer before anybody can sense it. And I can say that I've started my career, the first study we ever done was for early detection of an oncogenic mutation. I consider it the first days of precision medicine because we developed a crazy PCR method to detect KRAS mutation. We found that they exist in pre-neoplastic lesion in samples that appeared by the pathologist to be completely normal. That was 20-some years ago. More recently, yes, we are performing studies that are aimed to early to diagnosis. We have done one such study in pancreatic cancer by looking at serum from patients that we have analyzed at the level of exosomes and proteome, and led us to identify new markers that nobody have identified so far and nobody have used so far. And in order to validate this, we have joined forces with a very strong group that deals and works on pancreatic cancer in Oregon. And they, over the last 2-3 years, took one by one the markers that we identified in order to validate them. And they managed to validate 3 or 4 of those markers. We had all together about 7, 8, which is very significant. And it's very exciting because potentially we may be in a position that we can go to a blood test and identify a specific marker for pancreatic cancer that distinguish it from healthy and so on and so forth. So yes, we do it. The bar is much higher, and we're trying to address it as we can.
We have another question, and it is that you mentioned that RNF5 null mice have IBD symptoms, and that anti-RNA F5 antibody therapy eliminate the IBD symptoms. What do you think the antibody is doing? Is it promoting the RNF5 function somehow?
Yeah, so I need to clarify because what we have discovered is that the lack of RNF5 indeed resulted in animals that were super sensitive to develop IBD-like symptoms. But the reason for the development of the symptom is not RNF5 because RNF5 is not there. But because RNF5 is not there, another protein took over and became very abundant. That protein is S188. And we've mapped that using biochemistry. And the goal we set to ourselves is to see whether we can neutralize this S188, this one guy. And if we neutralize it, would we be able to block IBD? And that was the experiment. So we have administered antibodies to mice that were about to develop IBD, were already in the course of developing IBD, and we were able to block the development of IBD. We were very, very satisfied by that. So that would mean that there is a way to potentially limit the development of certain inflammatory disorders like that. But that raised another big question, and that is, remember the first part of the story that I told you, and that is that the same mice are very brave or capable of inhibiting cancer. So what will happen to their ability to inhibit cancer if you block that S100A guy? Would you be abolishing that capacity? And that experiment we actually have done, and we found that largely we did not abolish the capacity. There was a slight decrease, but largely these animals retain their ability to fight tumors. So there is a way to balance between the anti-tumor immunity and autoimmunity, and we hope we can do that by fine-tuning what exactly this gene is doing.
So our next question is regarding the study methodology for melanoma and whether that is similar to other skin cancers, like squamous or other types, basal, those that might be less studied or studied at all. And the question is are they, or since they are much less fatal, is that not as much of an interest?
Yeah, so yes, with all honesty, the community has focused on the melanoma because it has been a very tough one. And if we would have spoken now 10 years ago or 15 years ago, it was one of the most devastating diseases to which we had no therapy. Aggressiveness of melanoma is provided primarily by the propensity to metastasize, a tumor that metastasizes. If you don't catch it and remove it early, very hard to control. Today, we are able, and this has been the poster child, to halt the development of melanoma with immunotherapy, immune checkpoint therapy. And yet, it is still the more troublesome among the skin cancer. The basal cell and squamous cell carcinoma have different mechanisms. They are driven by different pathways. They are being studied, of course, not to the same extent because they are not as harmful. Basal cell, you remove and you forget. Squamous, you need to pay attention more. Squamous can develop to more severe, dangerous type of clinical indications. But the community largely focused on, and our lab has largely focused on melanoma because of the severity of the disease.
Okay, and our next question is, what are the challenges of transforming prebiotic formulas in mice to humans for reducing the tumor growth?
Excellent question. So this is the biggest challenge actually, and we face. Many laboratories have came up with suggestive bacterial cocktails that can be administered and can help solve problem. I don't largely believe in that. Prebiotics have some chance, but I also trying to be very careful, as you already noted about that. So we are trying to undertake another route, and the route is to find metabolites. So largely to find what is that the bacterial strains that we think are important secrete. And if you can identify what is that they secrete, which is largely metabolites and the type of metabolites, we will have a matter, we will have a reagent, we have something that we can actually test and verify activity across species. So we have been working for the last 3 years, and this just shows you how complex the story can be, to try and find out what are the metabolites that are secreted by these 11 bacterial strains I mentioned to you. And we haven't finished. We are using heavy computer simulation and other methods in order to address this question, with the notion that once we have hints, we'll test them. And if we are able to identify the metabolite is able to turn on the immune system and fight cancer, this is going to be the phase where I'll feel comfortable to take this further to human. We are not there yet.
So we have a question that came in related to cancer, and how do we get out of the whack-a-mole, where every patient has their own pathogenesis for the disease and hence needs their own individualized treatment? Doesn't seem that that might be very practical. And how many large subgroups are there, for example, melanoma, that one can attempt to bucket the patients into?
It's a very, very good question, and it's a very realistic, because this is exactly the situation that we understand it now. So if years ago we had a tumor and we considered the tumor to be one mass, today we know that this mass consists of many, many different masses and many small lesions of cells that are smart and waiting to take over when the time comes. So the heterogeneity of cancer is the biggest problem we are facing, and the biggest challenge we have. We do have technologies today that allow us to address this in a way that we were not able to years ago. Single cell technologies is one, where we're able to take the tumor and break it down to pieces and able to map the changes in the epigenetic and genetic trait in each of the piece. So we're able to see what makes this small lesion that is so different from others in the tumor. And then based on that, devise drugs or identify drugs that will hit not only the gross 99% of the tumor, but also the 0.01 that is likely to take over when the tumor has disappeared. The classic example is the example of melanoma that was treated years ago. And there was a picture that many of you have seen with a BRAF inhibitor. And the melanoma patient has full body covered with metastatic lesions, and they all disappeared within 6-8 weeks and came back about 2 months later, because they came back in the same spot because of resistance. So we have a heterogeneity that drives resistance, that drives metastasis, drive dormancy. These are the problems we now face. But I think that we will be able to overcome it, and the way we will overcome it by a computerized technology that allow us to get high resolution mapping of what happened in small lesion as opposed to the greater tumor. And based on that, a better understand how to fight those tumors in the small lesion as a whole. So I'm optimistic we are going to get there.
Great. Well, thank you again so much, Dr. Ronai, with joining us today and presenting on some of the research with your lab. We are excited and thank all of our attendees as well for joining us, and hope you learned something new. And we hope to see you again in the fall for our next Sunrise Science lecture series. Thank you all so much.
Thank you all. Thank you, Jen. Bye-bye.